So this is the basic mechanism of antigen recognition and immune response. So how antigen is recognized by our body, and because if your body is unable to recognize, everything depend on the recognition. If your body is not able to recognize the antigen, यानी अगर body पता ही नहीं कर पा रहा है कि if there is an antigen or not, because ultimately we have to differentiate between self and non-self, because everything that is not of our own body, that is considered as a non-self, and for every non-self thing our body has to generate an immune response. So for that our immune system should be able to recognize an antigen. So how they recognize it? So whenever there is an pathogen which is encountered in our body through any means, so we are having antigen presenting cells. We are having different APCs in our cell like macrophages, B cell, and also the dendritic cell. So we have different kind of cells which are regarded as the APCs of our immune system. So what they do, they take the pathogen, they process it along with the MHC. Without the major histocompatibility complex, we have different kind of MHC, MHC1, 2, and 3. So APCs along with the MHC, what they do, they process the antigen and they offer it to the B cell, because B cell is the only cell which will differentiate into plasma and effector cell, and they will then synthesize the antibodies. So this is the first way. But after this, this is the first way, but the second way is what, they can also give directly this antigen to the T cell. They will give signal for the T cell activation, and when T cell activates, they will start humoral response. Firstly, always humoral response initiates, and after that cells are involved. When cell will involve, if TC cell is involved, if T helper cell is involved, then your cell mediated immunity is active and they will start killing the cell. What they will do, they will start the phagocytosis of the antigen, increased phagocytosis. So there was an technical term, opsonization. There was a term, opsonization, right? So this is an overall overview, you can say, how our body recognize. So it can recognize directly by B cell or it can recognize by the T cell activation. So just a minute. Yes, this was the recognition part. CD4 plus T cells always recognize antigen on MHC class 2. CD8 always recognize antigen on MHC class 1. So I just told you that we have different kind of MHC classes. So class 1, MHC 1 recognize what? CD8 cell. And CD8 is present on cytotoxic T cell. Likewise, class 2 recognize C along with CD4 and it is present on T helper cell. ये 4 से you can remember. H looks like a, if this is removed, this looks like a 4. So this is the way you can remember. TH means CD4, TC means CD8. Now this is the most important part which we have to focus on, how the vaccine is developed in any of the pharma company, you can say any of the industry, you can say. So there are different phases which has to be processed for the development of a vaccine. So the vaccine development, the initial process is known as the preclinical stage. Why preclinical stage? Because it is still under research, it is still under investigation, it is still not fully approved by the FDA. So once it will be approved by FDA, you can say that this is market available vaccine. So the preclinical phase is conducted before testing in humans. So always we have term preclinical, clinical. So when you are talking about clinical, it means it has already gone through the animal studies and it has been found safe. It can be given to human subjects. So we are studying firstly the preclinical stage. So it will ensure the safety, immunogenicity, efficacy in vitro and in vivo. Of course, for first time when we do the experiment, we go for the in vitro one. Now, in vitro one firstly, or you can say firstly in vivo and then in vitro, cell lines, etc. So preclinical phase is the most important, you can say, or the crucial one. Without going into the preclinical phase, you cannot jump onto the direct human trial, because all it depends on the safety of the drug, whether it is immunogenic or not. Immunogenicity means whether it is able to generate immune response in a human body or not. Suppose you have developed a vaccine. So vaccine, whenever it is introduced into the body, it should be able to generate immune response. So if it is not able to generate the immune response, you can term this as a non-immunogenic. It means it is not capable of generating immune response. So this vaccine will not regarded as efficient vaccine, because ultimately your body should be able to recognize that antigen present in the vaccine, and it should be reacting against that. It should be providing the immune response, because this is the process. So you have to understand this one, how vaccine work in the body. So as I told you, whenever we are taking vaccine, it will immunize the person. Immunize the person means it will already have a adaptive memory that this antigen was encountered earlier. So whenever it will be introduced for the second time, it will offer different kind of immune response. So whenever a vaccine is able to do that, it is termed as an efficient vaccine. So immunization can be done by different part of the antigen. As I told you, you can take the subunit of the antigen, you can take the whole live antigen, you can take the weakened strain, inactivated one. So anything you can take as a uh, immunization model, or you can say as a vaccine. So after immunization, what your immune system will do? So your immune system will do what? It will start reacting towards the whatever antigen you have encountered in the form of vaccine. So it will retain the memory that already we are having this pathogen vaccine. So whenever it will come for the second time, your immune system will not take that much of time to react against it. So this is the overall principle of how vaccine works in our body. Now back to the preclinical stage. So now you should be able to learn that why preclinical trials are required. So the first step is antigen selection. So you can overall create a story that how the vaccines are developed. So the first one is you can see, the first one is exploration. So this exploration is generally it takes place in research laboratories. Like people used to find out that this particular whether it is herbal, inorganic, organic, doesn't matter. There is an item or substance which provides increased immunogenicity. So we start analyzing it preclinically. We try it in different animal models, and after that we after the clearance of the preclinical trial, we go for the human subject. So the first step is exploration, that this particular thing has a immunogenicity property, it is able to generate the immune response and all those things. So you should be able to know that key what are the different stages. So after the preclinical one, we are having clinical development. So this is the second stage, this is the third stage, and after this we are having, once it is approved by the regulatory bodies and once they have reviewed it, we are good to go. So you can write it down in your notebook. Just a minute. Okay, so these are the different steps. So third one is clinical development, and the fourth one is regulatory review and approval. So once your preclinical trial is good to go and you have also successfully passed the clinical trial, then you are having this regulatory review and approval thing. So uh, these are the steps which you have to take into consideration while designing the overall vaccine. Now, after that, we are having the manufacturing. Manufacturing means we synthesize in a very large scale, because we need vaccines for each and every individual. So we can design for the whole mass of population of that particular country or location, and after that we again check the quality, that whether it is up to the mark or not. So this is the different phase in which a vaccine is developed. So we're gonna study all these things in a very detail. So the first step in this exploration, you can say, or preclinical, both of these steps require firstly the antigen selection, because obviously we need a antigen which can act as a vaccine, which can act as a vaccine, because we know antigens, vaccines are what, vaccines. So uh, in this we decide that which part of the antigen can be taken, or also which antigen can be taken, like not only just part, which pathogen we have to take, which can elicit a strong immune response. Elicit means which can trigger a strong immune response. So anything that is capable of generating an immune response is chosen first, right? And if it is uh, not able to provide a strong immune response, then you can say that we have to choose a pathogen which is capable of generating an immune response, right? So first goal is to choose an antigen. So we always choose a antigen that have very high immunogenicity. Immunogenicity means it is able to generate immune response in our human body. अगर वो develop ही नहीं कर पा रहा है अपना immune response, then what's the point? उसको हम क्यों लेंगे? So this is the main goal while selecting the antigen, that it can be virus, it can be bacteria, but the thing is it should be very highly immunogenic. So this is the first goal. Now antigen can be of any type, so because vaccines are of different types, so you can take protein subunit of the vaccine, you can take the different what to say, different carbohydrate or polymer of any of the pathogen, like for example for SARS-CoV-2 we have taken the spike protein. In the polysaccharide, like for streptococcus pneumoniae we can take polysaccharide. Like any of the biochemical component of an antigen you can take as a vaccine development. So we are having protein subunit and polysaccharide, inactivated or attenuated pathogen, mRNA, DNA encoding the antigen, and virus-like particles. So these are the types of antigen or the part of the antigen which you can take. Now let me clear this slide. So the first part is in the preclinical phase we have to choose the antigen. पहले हम antigen choose करेंगे, और antigen भी कैसे choose करेंगे, which is very highly immunogenic, because what's the point, अगर वो immunogenic ही नहीं होगा तो हमारी body में जाके it will not provide the immune response, and for this purpose only we are taking the vaccine. So that is, there is no point if we are choosing a low immunogenic antigen. So this is the first case, and it is not necessary that you have to take the whole virus. You can take the protein of that particular pathogen, polysaccharide of that pathogen, or any of the mRNA based vaccine. It will be known as mRNA vaccine, DNA vaccine, or you can take any of the uh, a low virulence virus as a vector for coating your desired polymer subunit into it. That is known as the recombinant vaccine. So after this, we are having some criteria for the good antigen. Like you cannot take anything as an antigen to develop a vaccine. There must be some sort of criteria for the selection of this antigen, because all the success rate of vaccine or the efficacy of the vaccine depends on the nature of the antigen. So it should be highly immunogenic. Highly immunogenic, as I told you, it should be able to generate immune response very quickly without taking uh, time, that much of time. It should be surface exposed, secreted for easier recognition. Like suppose it should have some moieties, chemical moieties on its surface, so that it will be recognized by our different immune cells. Because if the what antigen really do, always they play hide and seek in our body, because they want to escape from our immune cells, because already in our body we are having blood all over the body, circulatory system is going on continuously, so they are circulating immune cells to each and every part of the body. So what the antigen do, antigen just modulate their outer layer, or they wear a shield like thing, a covering, which will not allow any of the uh, molecule immune cells to recognize them and to kill them. So we have to choose a antigen which is capable of having exposed chemical moieties that will be able to be recognized by the immune cell. So this is the requirement to be a good antigen. Now, conserved among strains, it should not be like it is heavily mutated, because whenever we are having a you can say unstable genome, it will start mutating every now and then. So we have to be very much careful while choosing the strain, because if it is continuously changing its genome, it will be difficult to target and to it will be very difficult to produce it in a very huge scale, because you have synthesized one vaccine. But if you want to synthesize for a mass production, then if its genome is continuously changing, then you will not be able to produce it in that efficient amount what you have done in your first stage. Now it should be associated with the protective immunity. This means it should not be able to produce any kind of you can say side effect in our body. Obviously we will have some kind of mild side effect, but the overall disease causing gene should be removed from this antigen, because otherwise it will cause you so much of harm. Now, what are the techniques for antigen discovery? So we are having different kind of techniques like the reverse vaccinology, proteomics and transcriptomics. And apart from this, we are having bioinformatics prediction and monoclonal antibody mapping. So all these techniques are very important because we are having requirement of all of these things. Now, reverse vaccinology is a computational based approach for the vaccine development, and what it uses basically genomics, genome based data and proteomics for the pathogen to identify potential vaccine target. So this method is very you can say a need of the hour, because in this we directly target the pathogen through different kind of genomic and proteomics approaches. So what we do, we just look into the different genomic and proteomics data. Like I will search for a particular pathogen X, I will search which gene of this particular X is responsible for causing any kind of disease. Let's suppose this X is a virus. Let's suppose this X is a virus. Its gene ABC is responsible for a particular disease. This is causing any kind of fatal disease. So I will extract out the genomic data, its sequence, and its proteomic data, its codons. And after that, what I will do, I will start uh, targeting this X as a antigen to develop a vaccine. So this is known as the reverse strategy. Firstly, we are going into the sequences, then we are targeting it, then we are preparing it, and then we are seeing it. It is reverse, यानी reverse है. पहले हम synthesize करके उसका application नहीं देख रहे हैं. We are firstly seeing the application, we are firstly seeing the genomic and proteomics data, and through that we are designing. So this is known as the reverse strategy. Reverse strategy, like if you are targeting any of the exams, so before preparing, you directly jump onto the whatever questions are asked in that particular exam, and then you will come to the theory. Yani hum generally conventionally what we used to do, we just read theory and then we come to the multiple choice questions. But in the reverse preparation, nowadays preparation, always I also recommend my student that always try to target multiple choice question, like any of the questions, and then come to the theory. So this is the case here, reverse vaccinology. Now we have proteomics and transcriptomics. Proteomics means protein based data and transcript means mRNA based data. So through this approach, we can all these techniques basically employs the computational biology, bioinformatics, you can say. So we have different bioinformatic tool for the for providing data of different level. So you can say we can use monoclonal antibody mapping. We can design a specific antibody for a particular epitope. Epitope is a antigenic determinant site. It is a specific site on an antigen that is recognized by the immune cell. यानी epitope क्या होता है? Epitopes are the specific site on the antigen. It is present on the antigen, and paratope is present on the antibody. So don't be confused. Epitope is present on the antigen that is recognized by the, suppose this is an antigen. So on its surface, there may be some chemical moieties, as the point, this one, surface exposed. So this is recognized by the immune cell. Immune cell. So these are the binding sites which are recognized or binded to the immune cell. So these are some of the criteria and the techniques for the antigen discovery. Now, as I have mentioned that we have different phases: exploratory, preclinical, clinical, and all these. So in the exploratory, what we do, this is research intensive phase, and you can say the pillar of the overall vaccine, because if the exploration is not there, how you will be having an idea that we can develop this antigen, this pathogen, as a potent vaccine. So this is a pillar of overall vaccine development process. So in this is very you can say time taking process and very research driven process, in which what we do, this is research intensive phase of the vaccine development process, which is designed to identify natural or synthetic antigen that might help prevent or treat a disease. So we firstly in this plan that we have some antigen which may be natural or which may be synthetic. So it can be developed as a vaccine. This thought had to be come into the mind of a researcher. Like in nowadays there are so many herbal based therapeutic option. So people are trying different uh, part of the plant as a phytotherapeutic drug. So this thought comes in their mind that this part of the, like for example in our lab we are doing the lantana camara. Lantana camara is a wild weed, you can say. So we are using its flower and its leaf for the for designing some therapeutic option for breast cancer, triple negative breast cancer. So that thought had to come in your mind that this thing has a potential, that it can be it's exploited as a potent or as a very efficient vaccine. So this is in the exploratory phase. Now, in the preclinical, what we do during this phase, researchers usually in private industries use tissue culture, cell culture, animal testing to determine whether the candidate vaccine will provide immunity or not. So, after your thought, like you have just thought that this tree leaf can be used as a vaccine. So क्या बस सोचने से हो जाएगा? No, you have to prove it experimentally through your research, through your statistically analyzed data, through different animal model testing, cell culture, in vitro, in vivo. You have to provide the data to the higher authority so that they will be allowing you to go for the clinical subjects, and then only your vaccine can be released after the satisfactory result of the clinical trial, after the regulatory bodies have given you the clearance and all those things. So after this thought, you have to do extensive research, you have to do a lot of experiments in vitro and in vivo to confirm that whether whatever you have thought that this leaf is antibacterial, anti-A, anti-O, so you will prove it experimentally. You will do different experiment, and after that you will be able to know that whether it is capable of providing immunity or not. So many candidate vaccine don't move on to the next stage of development because they fail to produce that immunity or prove harmful to test the subjects, because uh, इसी स्टेज पे पता चल जाता है कि ये जो वैक्सीन है इट इज useful or not, because if it is not capable of generating immune response in vivo cultures, in vitro cultures, so what's the point to take that particular antigen for the second stage? So you can say this is the filter stage. In this you will be, you have suppose 4 items, four antigens you are having, whether natural or synthetic, doesn't matter. So out of those four, after this preclinical trial, you are just with the result that only three is capable of generating a satisfactory immune response, and it meets all the criteria of a good antigen, this one. So you will take only this third antigen up to the clinical trial. So this is known as the filter stage. So whenever we go for any kind of product synthesis, we filter it out. If it is of no use, we will filter it out, we will not take it for the next level. So this is the case with the vaccine development preclinical stage in the overall development of the vaccine. Now, the clinical development. Once your drug is approved preclinically, the data are very satisfactory, and after that, what you will do, you will submit an application for an investigational new drug, that is IND, to the US Food and Drug Administration. As you all know, FDA is the largest and the uh, most crucial regulatory body which will decide you for every kind of food based product or any kind of pharma based product, whether it can be released into the market or whether it should be allowed for the clinical trial or not. So you will summarize all your findings, like whatever you are having, how you prepared your vaccine. You have to provide each and everything in a very detail, like the patent process. In the patent process, what we do, we provide every possible bit detail. Not, it should not be like superficially you have prepared the application, it will be rejected. So you have to prepare each and everything in very detail, like how you have synthesized the vaccine, through which method you have synthesized, and how much doses you have decided, how much animals you have taken in a trial, and every detail, how much it is efficient, what are the different biochemical parameters you have tested, and how much reduction or increase in the good or bad parameters, everything. Like, I'm just telling a bit of it, but you have to provide each and every detail. यानी कि मोटा मोटा तौर पे आपको सब कुछ बताना है कि क्या आपने यूज किया, कितने दिन तक का स्टडी किया, and also you have to provide the whether out of let's say we take generally 6 to 8 animals in each group, like control, some treatment, different doses you have, 200 mg or microgram or whatever be the dose. So we are having different kind of groups, treatment group and control group. So out of the 6 to 8 animals initially you have taken in your study, after the completion of your study, how much animals you have sacrificed. So all those detail we have to provide. So the FDA has 30 days to approve the application. Once the proposal has been approved, the vaccine must pass 3 trial stages of human testing. So this process is done when you are already having the data from your preclinical trial. It means I am having some kind of antigen. I have done all the in vivo and in vitro study. Now I want it to go through the clinical trial. It has been proved that it is performing very well in the in vivo cultures and in vitro cultures, and now it has to be trial sorry, it has to be trial in the human subject. So for that we are having 3 different phases again. In short, how to remember this? Uh, phase 1 means less than 100 persons. Phase 2 means hundreds of human test subject, you can say 100 to 1000, and this 1000 to 10,000, 10K you can say. So phase 1 means only a small group you will target, because you will not target that much, because you cannot take risk up to that much, because you don't know the effect of that particular vaccine on human subject. Obviously we are having similarity with the rodents, but not that much. You cannot say that 1% difference will cause a very fatal consequences. So it is very mandatory that you have to go step by step. In the phase 1 you cannot target more than 100 people, because then it will have some mass consequences. If it is having some ill effect, everybody will be in a trouble. So for that reason, always in phase 1 we will target the clinicians always target small group of people, like only 100 people. So after having a satisfactory response in these 100 people, they are entering into phase 2, like hundreds of human test subject, 100 to 1000. So after the satisfactory safety, then this candidate vaccine is safe, it can be explored more, it is not generating any kind of side effect. Like mild side effect will always be there. If it is not there, then the vaccine is not able to produce the immunogenic response, it's no point to use it. Always there are some mild side effects associated with each and every drug, despite people are saying that this is herbal with no side effect, like this, no, everything is wrong. Everything that is introduced into your body in the form of vaccine will always develop some milder symptoms, and it always depends on person to person. Suppose if any drug is uh, like provoking some major side effects in me, it is not necessary that you will also have the same kind of side effects. No, all it depends on our level of immunity we are having in our body. For suppose we are having the COVID-19 vaccine, so in so many people there was no such side effect, but in so many people there was high grade fever, body ache, and they are feeling lethargic. So I am one of them. So we don't have it depends on body to body. So in phase 2, as we have proved that it is safe, it is not that much of fatal consequences in 100 people, we can target up to 1k. So after having clearance or green signal or a satisfactory outcome from this one, now we can target the 1000 to 10 of test subjects, including the male and female. So it will continue to measure the safety, and after that it will finally provide you the effectiveness of the candidate vaccine, that this is the required vaccine, this has been proven very very efficient. After you have already read somewhere or heard somewhere that this vaccine is under phase 1 clinical trial, phase 2 clinical trial likewise. So after the phase 3 successful clearance of phase 3, it can be released into the market, but just after the phase 3 you cannot release into the market, you have to undergo again the regulation wala thing, regulatory affairs. You have to provide your everything to the regulatory affairs, and you then have to synthesize it on a very large scale, and again you have to go for the quality check. Because every time I have discussed that if you are synthesizing any kind of bioproduct, you have to go through the QC every step, because we cannot predict that only this step will spoil our product. Any of these step may spoil or degrade our vaccine, because obviously this is a chemical component. You cannot just say that this will be stable for years and years. No. So for every step we have the quality check, we assure that the quality is up to the mark and it can be consumed by the patients. So now this has been summarized, like phase 1, phase 2, phase 3, and phase 4 is what? Post marketing surveillance. So don't be confused. Mainly there are phase 1, phase 2, phase 3, and after that the post marketing surveillance, that what are the impacts after people are using it for a longer time, like the chronic effect of that particular vaccine is considered in the phase 4. So what are the participants? Only 22 healthy volunteers, 100 to 300 up to 1000 also. And in this, after 1000 to 10k or 3000 plus, or in this we generally take consideration into more than thousands. So duration may be few months, in phase 2 several months, 2 years, and phase 3 trials are generally chronic. You only have to provide, you don't have to provide only the acute data, you have to go through the chronic level studies. So ongoing, phase 4 are always ongoing because people are consuming it years after approval. Now focus, what will be the focus? Safe dose range. In this phase 1 we decide the dosage range. It means if you are giving to 100 people, you will decide the dosage, that whether 1 mg is safe to give to the patient or not. So in after this phase 1 you are having the safe limit of the drug, dosage of the drug. And in phase 2, as it is for up to 2 years to 3 years, you are having the immune response, whether it is able to generate the immune response or not, short term side effects, and this will be a long term side effect uh, in the phase 3, because this is for a very longer period, and this is also long term effect because already the product is released into the market and people are consuming it. So what are the long term effects? Are there any side effect? Now success indicator, so tolerability, dose identify, because in phase 1 if you are able to identify dose, because always the researchers try so many different drugs, like they decided through the MIC, IC50 and all those things. So they decide the dose, that this dose can be given to the patient up to, this is the tolerable limit of that particular healthy individual, so we can go up to this dosage. So in this you can be determine the dose of the particular vaccine whatever you have synthesized. So in phase 2, whether it is generating adequate immune response or not, and whether it is safe for whether it has any lethal side effect or not. And in phase 3 you are having statistically significant protection or improvement, because it is for the longer time. So after this you can with great assurance you can say that yes, this drug is very effective up to this, and these are the long term side effect, like you are feeling this, you are feeling weak, lethargic, and this. So uh, all these things are there. And in control group, we are control group are taken sometimes and sometimes it is not. And in phase 2 we are having standard and placebo. So placebo is what? It is a substance or a treatment process that appears real but has no therapeutic effect. So it is always taken into the clinical trial to compare the effect of an active drug, यानी जो हम ले रहे हैं उसके इफेक्ट को चेक करने के लिए, या यू कैन से कंपेयर द इफेक्ट ऑफ द एक्टिव ड्रग और द टेस्टिंग ड्रग with the control group. So basically placebo jo hai, it will refers to the positive changes a person will experience after receiving a placebo. So you can say that always we have to take the control. Without any control group you cannot go for the control uh, the comparison based thing, because people will ask you what you have taken in the positive control, what you have taken in the negative control, because in research if you are doing any experiment without control, and that too positive control, negative control, then you cannot say that my this treatments have this effect, because in always in a research we compare our every result with the control. So these are the things which are associated with phase 1, phase 2, phase 3. So this is the phase 1 is the basis for proceedings to the phase 2, because obviously if phase 1 will be satisfied, then you can go for the phase 2. If phase 2 is satisfactory, you can go for phase 3. And I told you after the phase 3 you have to go through the FDA and approval regulation wala thing. So in phase 4 what we do, it is already released into the market. So if any report is there indicating there is severe side effect after the consumption, so it is again refined, the whole preparation is refined, and then it is always again checked and rechecked that what are the preparation, what are the long term impacts on the individual consuming it, and the always they provide the risk. Again they update the risk that these are the things associated with this kind of vaccines. So these are the different phase 1, phase 2, phase 3 and phase 4 trial, clinical trial. Now regulatory review and approval. So as I told you, the first step was exploratory, second step was preclinical, third step was clinical, and after the clinical we are having some regulatory bodies which will review your application. So regulatory review and approval. If a vaccine passes through all the three phases of clinical development, 1, 2, 3, you have passed all, so the vaccine developer submits a biological license application, that is known as BLA, to the FDA. In emergency scenarios the FDA can grant for the use of unauthorized medical product. So this is the case when any such pandemic occurs. So all these procedures generally take very long time is required. Obviously there are so many different kind of clinical trial phases, like 1 year, 6 months, 3 to 4 years of clinical phase 3. So obviously it is required about 5 to 10 years, minimum of 5 years. So how you can take risk of up to the 5 to 10 years? So in case of COVID-19 also, we are having different kind of vaccine only in 1 to 2 years. So why this is so? Because at that time people are already in a very alarming situation. So what FDA do? FDA generally grants that you grant, they can grant the access that you can release your product into the market with certain guidelines, that you cannot target this group of people, like immunocompromised people. You can only target this, you have to provide this much of guideline in your product and everything. So this thing happened whenever there is an emergency situation. Emergency means severe pandemic is going on, so many severe cases. So in that case it is released, because already paperwork mein bahut sara time lagta hai. So COVID vaccine in use in the US state were given EUA, emergency use authorization. So always try to write all these terms whenever you used to read the newspaper or any Facebook post, LinkedIn post, whatever. So they already mention all these terms. So BLA is biologics license application, which you apply to FDA to release your product into the market. So कभी भी अगर emergency situation है, लग रहा है कि अगर हम लोग पेपर वर्क में ही फंसे रहे तो लोग तो इन्फ्लुएंस हो रहे हैं, जो भी हमारा पैथोजन है, लोग मर रहे हैं और यहाँ पेपर वर्क कर रहे हैं. सो दीज आर द थिंग्स गोइंग ऑन. सो उसे हटा के व्हाट एफडीए डू, एफडीए and all the medical related regulatory bodies urgently they just uh, give the green signal that you can release your product into the market because it is need of the hour. So emergency use authorization. So I just give the example of COVID-19. It has been regarded EUA, emergency use authorization. Now manufacturing. After the clearance of this regulation, regulatory bodies, you have to mass synthesize your product, because the vaccine should be distributed to each and every citizen of the country. So major drug manufacturer provide the infrastructure, personal equipment necessary to create mass quantities of the vaccine. They also reap the profit of successful or widely distributed drugs. So every uh, requirement is mentioned, like what are the different instrument infrastructure is required, labor, how many labors are required, and what are the you can say raw materials are required for the mass quantities of vaccine, and how many such phases are required to synthesize that vaccine. So all those things are written on decided on a piece of paper that is approved again, and then it is started then by the industries or the different pharma company. uh, they are having different kind of like tie up with different research regulatory bodies. So we will synthesize this much amount, we will synthesize this much amount. So through this way all the vaccines are widely distributed in a country. And after the synthesis, as I told you, again you have to go for the quality control, because you cannot say that your drug is safe for every step. So the approval and distribution is far from the end of the line. Stakeholders must adhere to procedures that allow them to track whether a vaccine is performing as anticipated. So before releasing into the market, you have to be sure, you have to give assurance that your vaccine is not causing any such fatal consequences. So these are the phase 4 trial optional studies that can be conducted following the release of a vaccine. So the vaccine adverse event reporting system and the vaccine safety data link are designed to monitor the performance, safety, and effectiveness of an approved vaccine. So see, there are so many steps. You cannot say these are the things which we are just studying, that this take this much amount of time. So everything has a some sort of, you can say, steps. We have to follow each and every step in order to get to the product. So there are so many sophistication involved in each and every process of vaccine development. You have to just, the first step is very much crucial, like the exploration. You have to explore so many things that has the immunogenicity property. So the first step and the crucial step is to find out the antigen. Second one is to go for the preclinical trial, and after that you have to go through different kind of clinical, and all these things have different uh, again different steps are involved in each and every clinical for example, if you are going into the preclinical trial, you are having different animal subjects, you will be having different animal subjects. So you have to take again the animal ethical committee approval. Without the approval of animal ethical committee, you cannot conduct any of the preclinical trial in your research. And for the clearance also, you have to provide every bit detail of it, how many animals you will use, how many animals you will sacrifice, because we cannot consider them as an animal. They are a living organism, they will be sacrificed for our research purpose. So we never use a term dead or like anything, we use sacrifice. So how many animal you will sacrifice, you have to provide the weight, approximately how much weight of animal or rodents you will use, 200 to 250 grams, 300 350 grams, and strain you have to provide, for how many days you will consider, and also you have to provide the side effect, that what are the some side effects. So in my case, in my masters, I have done a project in phytotherapeutics phytotherapeutics against the type 2 diabetes mellitus. So we have developed Tinospora, that is giloy, commonly known as giloy, very common herbal drug plant. So we have developed a nanoparticle using that particular plant against the type 2 diabetes mellitus. So for that, we have gone till preclinical trial. So we have already filed the patent, which is not yet granted. It is in the second step. So for that also, we have to provide all the things. Like I remember that I have at that time I presented uh, all my research plan in front of the all the members of the ethical committee. So at that time I was so you can say newbie, but I managed somehow with the help of my senior supervisor and all those. So you always have to go for the ethical whenever you are taking any organism for your human, like human, animal, you have to again go for the ethical committee clearance. So, and after that also, when you are involving any such human subject, you have to take informed consent, IC. Informed consent you have to take. In that informed consent, what you do, you provide all the details, and also you have to provide all the details in their local regional language, because people are not able to understand Hindi if they are from South, Tamil Nadu, Kerala, Chennai, and all those. So it is always recommended to provide information in a bit detail, and if possible in their own native language, because people will get to understand if they are not that much educated. So you will be uh, explaining them in their own local language, and you will be mentioning all the every detail. And also the human subjects are free to withdraw at any point of time. So they have basically the right to withdraw, right to withdraw. They can withdraw at any point of the study without even thinking of it, that what will happen to your study. Because if you are taking 30, let's say 30 subject human subjects, and if two of the patient is withdrawing from it at any point of the time, you will having only 28. Two voids will be created, but it will not influence their decision, because they are having the right to withdraw. And also they will be informed about the fatal consequences they may have after the consumption of the vaccine. यानी उन्हें ये भी बताया जाता है कि आपके में probably आपको ये चीजें देखने को मिल सकती है. Basically you have to reveal each and everything, because if you are hiding anything and if something major happens to them, you will be responsible and you will be having some ethical or you can say legal action. because you have been violated the ethical laws which are imposed on you. To you should have to follow all those things. So these are the things in which clinical trial goes through all of these things. You cannot say everything is easy. There are so many processes. You have to convince them. And also, if suppose you are giving them drug at 12 in the morning, like in the afternoon 12, so you have to just force them, or you have to just convince them to come at the correct time, because you are monitoring them till, let's say, 30 days, 60 days, 1 year, or whatever. So everything has to be taken into consideration कि उन्हें हमेशा आना है उतने टाइम पे जितने बजे ड्रग दिया है, and after that you, what are the different biochemical parameters you gonna check, and at what interval, like 2 months में check करेंगे, 1 month में check करेंगे, 15 days में check करेंगे, because you have to monitor each and every changes going on in the body of that clinical subject, because obviously you are taking you can say risk is there, risk is associated with each and every vaccine and any kind of product. So you have to monitor each and everything of that patient, so therefore it takes a lot of time to synthesize a vaccine. People usually say simply that itna time lag raha hai synthesize on mein, but these are these things which, as a biologist, you should always appreciate the efforts of each and every individual associated with each and every step of the vaccine development process. You can we can thanks the researchers, because they are the only one who starts everything, and after that we are going to the industry related stakeholders. They take the charge of some mass synthesizing and distribution, manufacturing, everything. So a lot of thing is involved in developing vaccine. So vaccine formulation, in vaccine formulation, what we do, we combine the antigen with component that enhances the stability, delivery, and immunogenicity. Suppose you have finalized the antigen that suppose किसी प्लांट का एक ड्रग है, सॉरी किसी प्लांट का एक पार्ट है, सपोज दिस इज अ लीफ, which you find that this is able to generate immune response whenever it is encountered into humans. So now in what formulation you will introduce it into the host, you have to decide that, obviously, because the leaf may have some bioactive component that may be very unstable, because the phytocomponents or the bioactive components of plant are very unstable. This is the problem while working with them. We have to stabilize them first, so you have to provide a coating so that it will ease the delivery. For delivery, most of the time chitosins are used, so chitosan delivery. And also, if you are coating it with anything, it should not be like it will decrease the immunogenicity of that antigen. It should always increase the immunogenicity of that particular antigen. So these are the things. Now component, antigen will be your component. Adjuvants, as I told you, it will enhance the immune response. So alum is the most widely and the commonly used aluminum salts are the since ancient time it is used as a adjuvant. MPLAs are what? Monophosphoryl lipid A. CPG is what? Cytosine, phosphate, guanine, oxy, deoxy, oligodeoxynucleotide, CPG island. So this is the common adjuvant which is nowadays used. Alum, MPLA, CPG. So basically alum means aluminium salts, aluminium hydroxide, aluminium phosphate. So the function is oldest and most widely used adjuvant, promotes strong antibody Th2 response. So basically why we use adjuvant? Because they are known to increase the effect of the whatever antigen or vaccine we are taking. So how they do it? There might be some mechanism. So they are known to trigger the antibody, that is Th2 response. And how the MPLA act? MPLA full form is monophosphoryl lipid A. This is the detoxified derivative of lipopolysaccharide. In gram-negative bacteria, we are having lipopolysaccharide layer outside the cell wall. So this lipopolysaccharide layer is always harmful. They are known to synthesize the endotoxin. So what we do, we just detoxify it. We remove the disease causing or the most toxic part of the LPS layer, and then we use this as a adjuvant. So this is known to bind with the TLR4. TLR stands for toll-like receptor. So it will induce the Th1 response. So this is the mechanism through which it will increase the immunogenicity of the whatever vaccine we are taking. Now CpG will do what? Synthetic DNA motif that mimic bacterial DNA. It acts as a bacterial DNA because in bacteria we are having multiple CpG nucleotides. So it will stimulate the TLR9 to activate B cell and Th1 responses. So we are having adjuvant to enhance the immune response. We are having stabilizers to prevent degradation, like we introduce sugar, we introduce amino acid into the antigen preparation. So stabilizers we add, preservatives we add because we have to increase the shelf life of the vaccine. It should be stable enough for months to years. So we use preservative, and for the delivery system we use nanoparticle delivery, liposomes, emulsions. So all these, this is a modern day delivery, and these are used since very long, liposomes and emulsion, because our body is having hydrophobic moiety, so it will easily in liposome we can easily introduce our vaccine, so it will be easily diffused in our body. Now types of formulation. So liquid formulation we are having, lyophilized freeze dried powder we are having, encapsulated in nanoparticle or lipid carrier when you are using liposome and emulsion. So we are having liquid भी हो सकता है, powdered भी हो सकता है, and it can be encapsulated into nano particle. Now after that we will decide the adjuvant selection factor, like we have to decide the route of administration, जैसे हमने जो भी पढ़ा था, muscular, orally, intravenously, and target of immune response, whether Th1 or Th2. Now safety profile, what will be the effect after administration of the vaccine into the host. So all these things are decided into the preclinical stages. So after this we will continue tomorrow because there is a lot to study. Obviously 2 to 3 slides are okay. We'll continue this tomorrow. So let me stop it and we will meet in the next class.